Hello guys, welcome to this YouTube channel. A good day to everyone watching this video. In this video, I'm going to discuss the answer key for the GATE Excel Biochemistry section of the 2015 paper. So let's get started. The first question. Which one of the following molecules, small molecules, is a prerequisite for fatty acid oxidation? So if you have read about fatty acid oxidation, the correct answer here is carnitine. That is because it acts as a carrier to transport acyl groups into the mitochondria for fatty acid oxidation. So the correct answer here is carnitine. So that's a pretty fact-based easy question. Moving on to the next question. Which one of the following bases is not found in the T arm of an amino acyl tRNA? So clearly it's the T arm, which means the correct answer here is dihydrouridine. So the dihydrouridine is exclusively found in the D arm of the amino acyl tRNA. However, the rest three, which is pseudouridine, uracil, and guanine, they are found in the T arm. So the correct answer here is dihydrouridine. So this is also a pretty easy question. You can also answer this by deductive reasoning if you know that there is a D arm that exists in tRNA and that D arm exclusively contains dihydrouridine, which is not present anywhere else. Moving on to the next question. Oxidation of one molecule of glucose via the glycerol phosphate shuttle produces. So the correct answer here is 30 molecules of ATP. So if you look at the different energy metabolites that are produced upon complete oxidation of one molecule of glucose via the glycerol phosphate shuttle, you need to take into account from glycolysis, then pyruvate dehydrogenase complex reaction, then Krebs cycle, and then oxidative phosphorylation, wherein the glycerol 3-phosphate shuttle will ultimately result in the entry of NADH into the mitochondria via FADH2 that ultimately results in the production of 1.5 ATP per FADH2 molecule. So if you do the complete maths of all this, like that includes complete energy metabolites production from glycolysis till this particular step, then the answer comes to 30 molecules of ATP. So just for the purpose of keeping this video short and not going too much into how the balance sheet is done for this, I'm just briefly going through different steps in the oxidation of glucose molecule and then telling you ultimately the end it is the glycerol phosphate shuttle which results in FADH2 producing 1.5 ATP molecule, and there is also another shuttle called the malate aspartate shuttle, and there it is the NADH itself produces 2.5 ATP per molecule of NADH. So these details have to be remembered when you calculate the final molecules of ATP. So moving on to the next question. Ribulose 5-phosphate epimerase is involved in which one of the following processes? The answer is pretty simple. It is the pentose phosphate pathway. If you look at the other three options, that is glycolysis, TCA cycle, and glycosylation, this molecule is not involved, and it is the pentose phosphate pathway that uses ribulose 5-phosphate epimerase in one of its reactions. So the correct answer here is pentose phosphate pathway. So this is a pretty easy question to answer. Moving on to the next question. Proteolytic enzymes are usually biosynthesized as large inactive precursors known as, the correct answer here is zymogens. So it's not holoenzymes and it's not ribozymes, which are catalytic RNAs, and it's not also apoenzymes without the active site molecules. So the correct answer here is zymogens, and if you could also remember some of your biochemistry techniques, zymogens are also used to actually remove the cell wall of certain cells. So that is also an important way by which zymogens are being used. And these proteolytic enzymes, when they are large and inactive precursors, they are called zymogens. So moving on to the next question. So the formation of a carbocation, also known as oxonium ion, occurs during the reaction catalyzed by, the correct answer here is lysozyme. So if you look at the mechanism of action of lysozyme, which is repeatedly asked in many biology competitive exams, then you will know that in the reaction catalyzed by lysozyme, there is a carbocation intermediate step. So I request everybody watching this video to have a good look at the mechanism of action of the enzymes. Three important enzymes that include lysozyme, ribonuclease A, and of course chymotrypsin. So these are pretty repeatedly tested. And for this question, the correct answer here is lysozyme. Moving on to the next question. So here, which one of the following amino acid substitution is likely to cause the largest change in protein conformation? So here you need to look at what class of amino acids is being changed to what class of amino acids. So if you look at the first option, phenylalanine is being substituted by isoleucine. So here it's non-polar to non-polar, so not a big change is expected. From serine through threonine, again it's from polar to polar, so not a big change is expected. From glutamine to tyrosine, again it's from polar to polar. However, if you look at option D, which is the correct answer, it is from glutamic acid to valine. So here it's a very big change in the protein conformation, and also this is, if I'm right, this is also the substitution that is seen in this deadly disease called sickle cell anemia, where glutamic acid is substituted by valine. So the correct answer here is option D. Moving on to the next question. Which one of the following does not constitute the lipid moiety in a lipid-linked membrane protein? So if you look at the structures of lipid-linked membrane proteins, you will find palmitic acid, you will find farnesyl groups, and you will also find myristic acid. However, what is not found in the lipid moiety in lipid-linked membrane protein is answer B, which is stearic acid. So you need to look at the basic structures of these biomolecules in order to answer these questions. So that's pretty simple and straightforward. Moving on to the next question. A closed circular bDNA of 4000 base pairs is negatively supercoiled by the introduction of 4 Brits. The superhelical density of the resultant DNA molecule will be. So you need to first note this fact that there are number of base pairs per turn in a bDNA is equal to 10 base pairs. So that is known. So the number of base pairs will be equal to 4000, the number of turns is equal to 4000 divided by 10, which is equal to 400 turns. And we know that the width is 4, and since it is a negative supercoiling, we have to take the negative sign into account, and hence the superhelical density is equal to negative 4 divided by negative, which is the number of Brits, divided by the number of turns, which is 400. So here the answer comes to be minus 0.01. So this is a pretty easy question to solve if you know that the number of base pairs per turn in a bDNA is 10 base pairs. So that's pretty simple. Moving on to the next question. Which one of the following is not a receptor tyrosine kinase? So this is a pretty easy question to solve if you know how different signaling pathways work and which receptors are involved in which signaling pathway. So that's a really big chapter to discuss, and just to keep this video short, the correct answer here is TGF-beta receptor. So this receptor is not a tyrosine kinase receptor. However, if you look at the rest 3, which is platelet-derived growth factor receptor, or insulin growth factor 1 receptor, or macrophage colony stimulating factor receptor, all of these are receptor tyrosine kinase. Phosphorylation takes place at tyrosine residues. However, TGF-beta receptor is not a tyrosine kinase receptor. Moving on to the next question. Match the entries in column 1 with those in column 2. So here, let's start with vitamin B1. So vitamin B1, of course, requires thymine pyrophosphate for its reaction. Carboxypeptidase requires Zn2+, which is zinc ion, for its action. TCA cycle involves the use of this enzyme called aconitase, which converts citrate to cis-aconitate and then to isocitrate. And then reducing sugar, which is lactose, which is a reducing sugar. So the correct answer here is option A. So this is pretty easy to solve if you know the basic facts of biochemistry. Moving on to the next question. So here, the following table provides information about 4 proteins. So here you have the native molecular weight in Daltons and also the isoelectric point, which is actually not needed, and then the type. So which one of the following option correctly identifies the order of elution in size exclusion chromatography and the increasing order of mobility in SDS polyacrylamide gel? So basically, let's first look at size exclusion chromatography. So in size exclusion chromatography, the order of elution is from large to small. So the large proteins will elute first, followed by the smaller ones. So, and it is based on the native molecular weight because you don't do any denaturation in size exclusion chromatography. So in that case, if you look at this, the S has the highest molecular weight, so S comes out first, and then it is Q, which has the next highest molecular weight, and then it is P, which has the next higher molecular weight, followed by R. So in size exclusion, so for size exclusion, the order of elution is SQPR, right? Yes. Now we need to look at SDS page. For SDS page, there is denaturation that happens, which means the proteins are no longer as dimers or trimers. So they only exist as monomers. So this one exists as a monomer with molecular weight of 32,000. This will move as a monomer, and this one it gets broken into two. That is homodimer. Since it's a homodimer, it gets monomerized. So this will be now each monomer would have a molecular weight of 20,000. Yes, and for the other one, it exists as a monomer. So that will again be 25,000. It will move as a protein of molecular weight 25,000. And next, this one exists as a homotrimer, which means you need to divide it by 3 in order to monomerize it. So that will be 15,000. And this one exists as 32,000. So in SDS page, the largest proteins or the proteins with higher molecular weight, they move slower compared to the smaller ones, which means the distance of migration in the increasing order will be from large to small. So in that case, again, P will migrate the slowest distance, followed by R, because that has the next highest molecular weight, followed by Q, followed by S. So the increasing order of migration distance is this in SDS page, and for size exclusion, the order of elution is SQPR. So the correct answer here is option D. So this is a pretty simple question to solve if you know that you need to divide here, and here you just have to remember from which protein I need to have the order of elution as the first one. So let's moving on to the next question. So the predicted molar extension coefficient at 280 nanometer for the peptide, so this is a really big peptide sequence that's given here, is watt molar inverse centimeter inverse, and they have also given the molar extension coefficients for phenylalanine, tryptophan and tyrosine at 280 nanometer. And the reason why they've given only for these three is that the absorbance that you're going to observe for the peptide at 280 nanometer is primarily because of these amino acids. So if you know the 1-letter code for these amino acids, then and you just have to basically add in order to get the answer. So for phenylalanine it is F, for tryptophan it's W, and for tyrosine it's Y. So from the given sequence, if you can find the number of phenylalanines, tryptophans and tyrosines and multiply them with the respective molar extension coefficients and add it, the answer is going to be for the entire peptide sequence. So the number of phenylalanines is 4, the number of tryptophans is 2 from the sequence if you just count, and then the number of tyrosines is going to be 3. And for each phenylalanine you have 200, and from each tryptophan you have 5,600, and from each tyrosine you have 1,400. So 4 into plus 2 into plus 3 into, the correct answer here is 16,200. You basically do the addition. So this is a very pretty simple question to solve. Moving on to the next question. So here, match the contents of column 1 with the most appropriate options in column 2. So here, complement C1Q, it binds to the Fc region of the antibody. Yes, and L-selectin binds to CD34. And membrane attack complex is usually from the B part of the complement protein, so it's complement C5b. T helper cells have CD40 ligand in them. So the correct answer here is option C, which is P3, Q1, R2 and S5. So that's an easy question to solve. Usually the CA1s of the complement proteins are involved in producing inflammatory reactions like anaphylaxis as well. Moving on to the next question. So here, the value of delta G at 37 degrees Celsius for the movement of Ca2 plus ions from the endoplasmic reticulum, where the Ca2 plus concentration is 1 millimolar, to the cytosol, where the Ca2 plus concentration is 0.1 micromolar, at minus 50 millivolt membrane potential is watt kilojoule per mole. And they have also given the constant, that is R, gas constant, and the Faraday. So the formula for delta G is equal to NFE cell plus RT ln K. Okay, so here N is equal to 2 since it's calcium ion, so the valency is 2. F is Faraday, so it's 96,500 into E cell here is minus 50 into 10 power minus 3 plus R, which is 8.314 into T. In Kelvin it has to be because everything is in Kelvin. So 37 plus 273 would give you 310 when you change from Celsius to Kelvin, into ln K, which is product by the reactant. So the product concentration here is 0.1 micromolar. So it is better if you, let me just write it here. This is pretty towards the end. So 8.314 into 310 into ln product, which is 0.1 micromolar. So multiplied by minus 3 into 10 power minus 3 because the reactant is in millimolar. So that will be 1. So if you do the complete calculation for all of this, you get the final answer to be approximately minus 33.392 kilojoule per mole. So make sure that you take everything into account, and when you are converting ln to log, make sure that you multiply it by 2.303 and do the calculations, and make sure that the negative signs are not left out. So if you do all these things, you will get the answer to be minus 33.392. So just make sure until what decimal place do they expect this answer. So that's pretty simple. Moving on to the next question. So here they have given you a peptide, a small peptide, a dipeptide I would say, and here they ask you about the names of the different angles when you plot them on the Ramachandran plot. So which one of the following identifies the correctly matched pair? So here in the Ramachandran plot you mainly have two main angles, that is psi and phi, and apart from that here you also have Z, and which is omega, and also chi. So here the correct answer is option B, where W is 1. Yeah, W is 1, that's correct. And X is 3. So X here is this angle. So that corresponds to the phi. And W, which is this angle between C alpha and C, that is psi. And if you look at Y, which is 4, which is omega. So Y is here. So it's between the C and N. And Z is chi, which is 2. So this one is chi. So the correct answer here is this. So I request everybody watching this video to learn how the Ramachandran plot is plotted, and most importantly, which angle corresponds to which rotation, which rotation between which two atoms, basically the bond rotation between which two atoms in a peptide bond. So moving on to the next question. So here, which of the following statements is or are incorrect about hemoglobin? So hemoglobin demonstrates higher oxygen carrying capacity compared to myoglobin. So that is a correct statement. There is a covalent bonding between the 4 subunits of hemoglobin. So that's a wrong statement. During deoxygenation, the loss of the first oxygen molecule from oxygenated hemoglobin promotes the dissociation of oxygen from the other subunits. So this is also correct because it's cooperative binding as well as while releasing. So the correct answer here is option A. That's a pretty simple question. Moving on to the next question. So here, a 1.2 kilo base per DNA fragment was used as a template for PCR amplification using primers P1, P2, P3 and P4. So here you have 4 primers as you see in the figure, and the annealing positions of the primers of the template are also indicated by the numbers, and primers P2 and P3, they contain a point mutation. So each, this is indicated by the triangles. So PCR was first performed using the primer pair with P1 and P3. So when PCR was performed using this primer pair, so you get this region to be amplified. And however, if you perform a PCR in another vial, PCR was performed using P2 and P4. So here you get you get the amplification between this. So this region gets amplified, and the purified PCR products from both the vials, now you have this amplified product and this amplified product. So when you now mix both of them into one vial and you perform another round of PCR with P1 and P4, what will the final PCR product compose of? So here now you're going to have these two, and now you're going to have the PCR reaction for this and this. So in that case, you will have a 1.2 kilobase pair DNA with 2-point mutation, because the original DNA fragment is 1.2 kilobase pair, and however the amplified products, they already have 2-point mutations, and hence you will have the complete amplification of this entire DNA fragment with 2-point mutation, because there's already an overlapping sequence. So they will anneal, and then you will add the entire PCR reaction for the entire 1.2 kb DNA fragment, but with 2-point mutation. So that's a pretty simple question. Moving on to the next question. So here a cell suspension was subjected to a membrane disruption followed by differential centrifugation to fractionate the cellular components. Match the centrifugal conditions in column 1 to the appropriate subcellular components in column 2. So here, as you can see, the G also increases, which is the relative centrifugal force, and the time of centrifugation also increases, which means as from P to S, if you go, the size of the organelle that you want to separate should also decrease, because you need more centrifugal force and more time to spin if the size of the organelle is going to be small. So in that case, the correct answer here is option A, which is this, where the nucleus is the biggest one of the 4 organelles here, followed by lysosomes and peroxisomes, followed by microsomes and small vesicles, and the ribosomes, which are the smallest, they require really high relative centrifugal force for a lot of time of spinning, So the correct answer here is option A. Moving on to the next question, or the last question. So here, this is a pretty easy question. So let me just zoom in here. So given below are the maps of 1,200 base pair long DNA insert. So this is the insert. And the 3000 base pair expression vector here, the BAM-H1B, so B is for the BAM-H1 restriction enzyme site, and the HIND3 restriction sites, which is by H, and the DNA length between them are indicated in base pairs now. So the insert is cloned into the vector at the BAM-H1 site, so that basically the insert starts from here. This is very important to know. It's not from here, but rather from here, and the desired orientation is also shown by the arrow. So after cloning, the orientation of the insert in the recombinant plasmid is tested by complete HIND3 digestion, followed by agarose gel electrophoresis. So that's what you basically do. Which of the following band patterns reveals the correct orientation of the insert in the construct? So first of all, you should know how the insert is going to look like in the plasmid. So this is the plasmid that you have. So here the plasmid already has one HIND3, and there's also another BAM-H1 site in the plasmid already. So now the insert is going to start from here. So which means there is one HIND3 here. If you look at the insert, so let me just draw the insert here again. So if you look at the insert, there is BAM1, BAM-H1 and HIND3 already separated by 300 base pairs here, followed by another HIND3 here, which is I guess 200 base pairs, and then here you have another B, BAM-H1 site, so that has 700 base pairs. So if you look at this, I just drew it from the BAM-H1 site. So this is going to be the insert, and this is going to be the rest of the plasmid. So the rest of the plasmid here corresponds to, I guess it's going to be 3000 base pairs as shown in the question. Yes, perfect. So from this to this, it's going to be 3000 base pairs. Okay. So now we are going to do complete HIND3 digestion, right? So that was given. So when you do complete HIND3 digestion, so this part is going to get cleaved. This part is going to get cleaved, and here also you have HIND3. Yeah. So this part is also going to get cleaved. So when these parts are going to get cleaved, so here what you have is, you have 300 base pairs plus 300 base pairs, so that is going to be 600 base pair fragment that is going to be created. And here you have a 200 base pair fragment that is going to be created. And here you have 700 base pairs that is already there, but then you need to take from here to here. So that is going to be, so 700 base pairs plus 3000 minus 300, because this 300 is removed by hint 3 digestion. So that is going to be 3400. So the fragments that you basically get correspond to 600 base pairs, 200 base pairs, and 3400 base pairs. So if you look at the options here, so this clearly corresponds to option A. So in option A, you have the migration here is 3400 here approximately, here you have approximately 600, and here it's approximately 200. So with that, I'm completing the discussion for the answer key for the biochemistry section of GATE Excel 2015 paper. I hope you found this video interesting and useful. I wish everybody preparing for this exam best of luck, and hope you all ace the exam, and then hope you all approach all the questions logically. Thank you and have a nice day.